Okay, I would like to welcome Andrew to talk to us about some exciting things about the lifespan and mutation, why elephants don't get cancer. And I would like to say hello to all our students and the participants who are there, who unfortunately we don't see. And I'm sure most of you have heard about Andrew. He's been coming to Sabanci University for the last 20 years, I think. Is it 20 years?
Slightly more, Zara.
Even more, my God, no. So, he's currently at Harvard University. He's an evolutionary biologist and a historian of science. And he'll tell you more about himself if he wants to, or if you ask him questions. So, Andrew, please go ahead.
Thank you, Zara. I'm not going to tell you more about me because this isn't about me. It's about elephants. Well, it's not about elephants. It's about all sorts of things. But elephants are, shall we say, the headliner. Yes, I am tragically here. So usually at this time of year, I am on campus at Sabanci, but not this time, tragically. So this is Harvard University. And let's be more specific. This is the building that I work in. It's called the Biological Laboratories. And in fact, my office is just behind the window there, arrowed window on the left. So that's where I am now. And the reason I highlight this is partly because I love this building. I think it's a really nice piece of between the wars design. But I'm talking partly about elephants. And if you zoom in here, we can see that in the frieze work and the brickwork in the top of the building is this fantastic montage of a herd of elephants. So despite the fact I'm not in Africa where there are elephants, despite the fact I'm not in Turkey where you are, I'm in a good place from an elephant point of view because they're plastered over the outside of this building. But I would much prefer, obviously, to be in the most beautiful city in the world. And I'm very sorry not to be joining you there. I hope to be there soon, regardless. Not only is it beautiful, but it also has the best food in the world. So that's another rather serious reason for being there. Okay, let's do some work. Let's talk about lifespan, mutation, and eventually elephants. But let's just start sort of globally with from an evolutionary perspective than anything else, the issue of lifespan. So how long will you live? Well, as you know, there are slightly different depending on country, depending on sex and so on, lifespans for humans. And you can gauge it yourself if you fill out this form, which asks you a bunch of rather inane questions about whether you smoke and whether you exercise, whether you're overweight and so on. This is not a scientific survey. It's actually produced by an insurance company. They're the ones who are interested in how long you live because their profit margins stand or fall by how long you live in terms of life insurance. So you can, well, don't do it now. You can calculate how long you're going to last on this basis. But let's look at some other species. The longest lived organism is a rather drab mollusk. So I shouldn't say organism, animal, a rather drab mollusk, which lasts over 500 years. A Greenland shark lasts 400 years. These are all the real long-lived species. All of these are over 100 years. Pretty impressive, actually, if you think about it. But obviously, there are plenty of species which last, certainly as adults, think of a butterfly or a mayfly, which lasts literally a few days. So what are the determinants of lifespan? Well, it's a very straightforward body of evolutionary theory that addresses this. The argument is basically this, that if you've got a deleterious allele that affects you early in life, before you reproduce, and natural selection will knock that out of the population, then that's eliminated. But if there's an allele, a mutation, a deleterious mutation, which say results in cancer or whatever, that acts late in life, after you've reproduced, then it's not going to get knocked out of the population, right? And a canonical example is this horrible disease, Huntington's disease. So this has a simple monogenic genetic basis. It's a known mutation, runs in families, but it's late onset. So you don't know whether or not you are you have the allele until after you've reproduced. Okay, fine. But it still begs the question as to where a species lifespan comes from. And it comes from what I'm calling extrinsic factors. So these are the things that can go wrong. Now let's think back to ancestral humans. On the plains of Africa, you could be eaten by a lion, you could die of a disease, not COVID, but a prior epidemic. You could be struck by lightning, whatever. Things go wrong, you die, okay? And there's a reasonable chance in the ancestral environment that you'll almost certainly be dead, courtesy one of these unhappy events, by, say, the age of 30. So what does that mean? So take the scenario I've just given you with early alleles not being selected out. If they affect you before the age of 30, then natural selection removes them. If they're things which affect you after the age of 30, you're already dead. So natural selection doesn't care. So that's where we are now. Maybe our ancestors all died by 30 or 35, in which case there was no selection during that phase of human evolution against the mutations, against the deleterious alleles that killed you after 35. And so that's what kills us now. Those are what I'm calling the intrinsic factors: heart disease, diabetes, all these modern killers, intrinsic factors. But this still doesn't really address a really interesting issue. Why do we get these vast differences in lifespan among species? Give you my favorite example. You take a rat, it probably lasts between 2 and 4 years. If you take a squirrel, which is basically a rat that lives in a tree with a fluffy tail, they have a life expectancy of 25 years. So basically almost an order of magnitude longer. Why? Well, it comes down to the environment in which the species evolved. And I'm going to illustrate this in a slightly weird way, an example that Peter Medawar, a famous immunologist, used to illustrate this idea. We have here 2 bars. This is the bad bar. It's rowdy, it's dangerous, people get into fights all the time. And this is a good bar where there are lots of ferns and everyone's very agreeable and well-behaved, okay? So we've got a good environment and bad environment, and we're going to start off, and what we're interested in is the longevity by extrinsic factors. Extrinsic factors are the factors that break beer glasses, the longevity of beer glasses, okay? So obviously, in the bad bar, these glasses are going to have a shorter life expectancy than these glasses in the good bar. And we can actually see that in action. Whoa! 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 So we've lost 3 in one night here, and only 1 in the other one. Let's keep going. So look, in 2 nights, we've killed off all the poor old glasses in the bad bar. We've killed 1 in the good bar. Now, let's think about this. What does that mean? That means that these 2 environments have very different survivorship curves for the glasses. If you're in a good environment, the good bar environment, you're going to last quite a while. Whereas if you're in the bad bar environment, you're going to get trashed very rapidly. So what does that mean? Well, in the case of the bad bar environment, there's no selection against deleterious mutations, which say affect 3-day-old glasses. Whereas, of course, in the good bar environment, there is strong selection against the deleterious mutation that affects a 3-day-old glass. You get the argument. It depends on the extrinsic factors of the environment. That's going to govern your expected lifespan. Okay, that's fine. So we understand in very broad terms the evolutionary factors underlying differences in lifespan. Now let's look within a species and see what differences we can find. Look, the most obvious one in our species is sex. So these are different populations across the planet by nation. And this is male life expectancy, female life expectancy, and here's the fall line where male equals female. Everything is north of that line. What does that mean? You know what it means. It means that females in humans are universally, on average, longer lived than us poor, sad males. Now, why is that? All sorts of interesting, and some of them are sort of obvious ideas. Men go off and fight in wars or get into fights and generally are more sort of risk prone than females and so on. And obviously these factors play a role. But I think the key thing here, which is a really interesting but really simple genetic idea, is the differences in sex chromosomes between females and males. Males, as you know, are XY. We call that the heterogametic sex because they've got two different sex chromosomes, hetero-different. The female has two X chromosomes, homogametic, the same, two of the same sex chromosomes. Now, what that means, this is a critical difference, because the Y chromosome is pathetic, it's tiny. So effectively, the male has one copy of the X chromosome information, the female has two. Now think back to introductory Mendel. If you've got a deleterious allele, a bad mutation, is probably recessive because it probably breaks something. So you need two copies of that in a typical diploid situation. So in the female, often if the female is carrying a recessive allele, she's got the good copy as well, so it's masked. So it doesn't affect the female. The male doesn't mask it. So the male is going to express all those X-linked deleterious alleles, which is why typically across mammals, females are longer lived than males. But you're going to say, well, what about the other arguments? Well, let's look at the sort of natural experiment, which is kind of cool. In birds, the chromosomes are flipped. The males are homogametic. They have two of the same chromosome. And the females are heterogametic. They have two different chromosomes. Okay, so exact flip of the situation. Who lives longest in birds? Males do typically. Okay, so there you've got a flip in the genetic system. You get a flip in the demography as well. And you can look at this on a very broad scale. This is a recent paper that looked at the effect of hetero versus homogamy in sex chromosomes on longevity. Simple analysis. This is the family tree of all these groups, and this is a lot. We've got monkeys and humans up here. We've got birds down here, insects down here. There's a lot of species, and the line is where males and females have the same lifespan. Okay, and on the right is where the heterogametic in mammals, that's males. In birds, that's females. Have the longer life, have the shorter lifespan, sorry. And on the left of the line is where the homogametic in birds, that's males, and in mammals, that's females, have the longer lifespan. Notice the enormous skew to the right here. So this is a pretty general pattern that if you've got heterogametic sex, it's a heterogametic sex, which is going to be the one which has a lower lifespan than the homogametic sex. Okay, interesting general result. The other factor, and I'm going to be talking about this a fair bit in the rest of the talk, that pertains to survivorship within species is size. And here's a classic data set, dog breed size. These are all one species, right? Even if they're kind of crazily disparate from a Great Dane to whatever this sad little yappy thing is, whatever the difference is, a major determinant of the longevity of a dog breed is its size. And the bigger you are as a dog, the sooner you're going to die. And why is that? Why is big a disadvantage? Well, here we get into the delights of cancer. As I'm sure you know, cancer is a product of dysregulated, uncontrolled, chaotic, anarchic cell division. And what typically happens in the start of cancer is there new mutations occur in the individual that damage the apparatus that control regular cell division. Okay, so the top, you've got a tumor suppressor gene. So there are a whole suite of genes which basically control the cell cycle. And if they see something going wrong, they'll intervene, or the gene product will intervene and cut off that cell lineage. Okay, so these are the policemen, if you like, of cells. If you have one mutation which knocks out the function of one of these genes, you're probably okay because you've still got an active copy. But if you have two mutations, so if you've now got a homozygote, and as I say, this is happening in you, this is somatic mutation. So, or you might have inherited one copy from your parents, and then you get a somatic mutation which knocks out the second copy in your liver, let's say, and you've got liver cancer, okay? That's one kind of cancer cause. The other are so-called proto-oncogenes, which mutate to become oncogenes. These are genes that promote cell division, and they should do it in a nice, controlled, orderly kind of way, but occasional mutations or regulatory effects will cause them to lose control. And so that's another mutation. Usually, these are effectively dominant, usually, because you only need one, because now you're producing too many cells. The cells have gone crazy. All I'm really saying here, and this is, I'm not a cancer expert by any stretch of the imagination, what I'm saying here is it's mutations that cause cancer. And here's a classic cascade. So multiple mutations occurring. This is in colon cancer. When and how do these somatic mutations arise? Well, there are all sorts of causes of mutation. Obviously, if you stand in front of an X-ray machine for too long, that's going to cause mutations. If you eat mutagens, that's going to cause mutations. But probably the single most common source of mutations is just errors in replication. Think about it. Cell is dividing, has to replicate that 3.5 billion base pairs of the genome. No, in fact, it's replicating two genomes because it's got both the maternal and paternal chromosomes on board. So that's a handy 7 billion base pairs for each cell duplication to replicate. That's a lot of replication, and we're pretty good at it, to be honest. Well, cells are pretty good at it, but they make mistakes. And it's mistakes that are mutations. And if the mistakes occur in the wrong places, say this is an accumulation of mutations going from normal colon tissue to pathogenic malignant colon cancer. And as I say, the single most common cause of this is your common or garden replication errors. So now, the fact that big dogs die more efficiently, shall we say, than small dogs is beginning to make sense. And actually, here's a data set looking at different breeds of dogs and the probability of them dying of cancer, okay? It's not beautiful, but you've got a strong positive relationship. The bigger you are as a dog, the more likely you are to die of cancer. Now, and why is that? Well, think about it. If you're a small dog, you don't have that many cells. Well, you still have quite a few cells. But if you're a big dog, it's not like big dogs have bigger cells. They just have more cells. What does that mean? More cell division. What does that mean? More potential for mutation. Ha! You say, I don't care about dogs. What about people? Same effect exists in people. So this is a survey of men in London. And this is kind of a cool piece of demography. We've passed them out by height. These are in inches. So that's what, 5 foot 6 inches, 5'9", and so on, 6 foot tall. And what we're looking at is the rate of cancer-related deaths. Now, if they're smokers and they're dying of lung cancer, there's no effect because a tall person is as likely to smoke as a short person, and smoking is the major determinant here, right? But if there's no smoking and we're looking at cancers unrelated to smoking, what do we see? Again, same increase in height from 5'6" to over 7 foot. You see a clear trend. You're more likely to get cancer unrelated to smoking if you're tall, more cell divisions than if you're short. Ha! You say, that's men, useless men. Happens to women too. So this is a similar data set. This is from the Million Woman study. And now we've gone metric. We've got height in centimeters. And what we have here is RR, which is a risk of cancer. So it's a similar metric. It's been normalized for the lowest group. And again, look, the straightforward increase. The taller you are, the more likely you are to get cancer. And this has been repeated by multiple studies. So here's another more recent study. This study confirms the positive association of height with risk of all cancers. So we've got an interesting problem here. This is happening within a species. The bigger you are, the more likely you are to get cancer, okay? But you've got small species, big species, medium species, and so on. Does this pattern hold up across species? In other words, if I'm a big animal, am I more likely to get cancer than a small animal? And in principle, yes. Because again, we've just said that mutations are introduced by cell replications. Big animals are going to have more cell replications than others. So it's actually kind of compelling. This is an analysis looking at 3 species, humans, elephants, and whales, looking at the probability of them contracting cancer by a given age of 70. And basically, the message here is that if everything was just extrapolated from the rates we observe in humans, then elephants would all get cancer and whales would all get cancer. End of story, right? But the interesting thing, and this is the interesting thing, is they don't. So in other words, the pattern that we see within species, bigger individuals be more prone to cancer, is not replicated across species. This is a really cool pattern. It was first identified by this British statistician man called Richard Peto, and it's become known as Peto's paradox, which is large animals, given the logic I've just given you, more cell divisions, more potential for mutation, more, therefore, surely cancer. Large animals should be much more cancer prone, but they're not, or at least they don't seem to be. And it turns out that assaying the relative probability of dying of cancer in animals other than our own species, we're easy. We just count the number who die in hospitals. How do we do that for humans? It wasn't until last year, actually, a big paper came out in Nature, which looked at, I think, 110,000 zoo animals and assessed their cancer risk. And so details are completely unimportant. This is the family tree, and the red bar is an indicator of cancer probability. There is some phylogenetic difference. As you can see, there's fewer in artiodactyls than elsewhere. They also did more analysis. You're more likely to get cancer if you eat meat, for example. But look, the key thing here is that cancer happens basically to every mammal that we've ever looked at. So cancer is a general mammalian phenomenon. So it's not like it doesn't happen in whales and elephants. The question now is still there. Why don't elephants and whales have cancer at a much higher rate? Well, so here's the paper. Don't worry with the detailed abstract. What I want you to take away from this is this message. We provide unequivocal evidence for the body size and longevity components of Peto's paradox, the paradox being that whales and elephants should have cancer all the time, but don't, by showing that cancer mortality risk is largely independent of both body mass and adult life expectancy. So in other words, being big is not a predictor of getting cancer. That's across species. It's true within species that being big is a predictor, but across species, because different species have evolved to accommodate different life cycles, life histories. So now let's get down to the nitty-gritty. What about mutation? Let's go and measure mutation, because that's ultimately what we're interested in. 2 ways we can do this. The first way is the sort of easy way, and that's measuring what we call germline mutation. Germline mutation is the mutations that occur in the production of gametes, sperm and eggs, such that you're going to have differences between junior and mum and dad. So this is a paper from 2017. And it's kind of cool. This is a simple exercise. Sequence mum's genome, sequence dad's genome, sequence junior's genome, and spot the difference, okay? And it turns out what we're interested in are sometimes called de novo mutations. It turns out that about 70 or 80 new mutations occur in the generation of every new human being, obviously with some variation on that. And notice too, and they're separated here by sex. So this is the new mutations that Junior has derived from dad, and these are the new mutations Junior has derived from mum. Notice two interesting things. One, you're getting many more mutations from dad. Notice these are single base pair changes from an A to a G or a C to a T or whatever, not big chromosomal changes. Mums have problems with late in life, think trisomy 21, for example, with chromosomal changes late in life, but then they don't supply a lot of mutations, and it doesn't vary much with age, as you can see. Whereas males dad produces many more mutations, and that keeps on going, because remember, this is menopause, mum stops reproducing, dad, think Rupert Murdoch, keeps on going and going and going, right? And Rupert Murdoch is just creating more and more mutagenized children, is the point. But why is there this difference between males and females? Women are born with all their eggs, okay? Yes, they're in a state of suspended animation and they're gradually developed through the menstrual cycle through life, but that's basically, the DNA replication has been done a long time beforehand. Men, on the other hand, are constantly undergoing cell division to produce ever more gametes, more sperm. So the difference here is lots of DNA replication, and I've just said that's the major source of mutation, not very much DNA replication at all. So that's interesting, but it's not really germane to what we're interested in. There are almost certainly going to be different factors affecting rates of mutation and the repair of mutation in gametes than in somatic cells. And it's somatic cells that we're interested in. These are your body cells, the liver cells that undergo mutation and become cancerous, the lung cells that undergo somatic mutation and become cancerous. So let's ignore findings on germline mutation and think about somatic mutation. Sorry for the huge slab of text, but this is a really cool paper. Only came out last year. And this is the first sort of canonical study across animals of rates of somatic mutation. That's actually a rather difficult thing to measure, and perhaps the sort of biggest triumph of this paper was the sort of technological achievement. Obviously, we've got to sequence lots and lots of DNA, but what we want is to get some metric of the accumulation of new mutations over the course of a lifetime of an individual. So we want to have, ideally, the descendants of a single cell, and they've produced many, many descendants over the time. And then we can go and sequence the descendants and figure out how different they are from the progenitor cell. They did this, and the details are important. They did this using a certain feature of the intestine. They're called colorectal crypts. That's a sexy name, isn't it? The key thing is each one consists of about 2,000 cells derived from a single cell. So by sequencing all those cells, you can get a handle on the differences which have accrued since that original progenitor cell originated. And the results are really cool. So here we've got 4 different species: dog, human, mouse, and of course the naked mole rat, the world's ugliest organism. And what we've got is on the x-axis, we've got age. Now, obviously, you've got hugely different ranges here, from up to 14 years in dogs, that's an old dog. 80 for humans, that's an old human. In mice, you're lucky if you beat 2 years. Naked mole rats remarkably live a very long time. So we've got huge range in x-axis scales. Notice we've got a basically linear relationship, however, with the accumulation of mutations. And what I want you to notice is that there's a bit of difference, especially with the mole rat. But what we've got is, by the time a human is 80, they've accumulated what, about 4,000 mutations. By the time a mouse is 2, it's accumulated half as many of those, 2,000. And a dog gets to 3,000 in its 14 years or whatever. In other words, and this is really amazing, the rate of somatic mutation is inversely correlated to the lifespan of the species. So what does that look like? So here we have a plot of lifespan. Here are a bunch of these species. The longest lived they have in their sample is humans. The shortest they have is mice. And this is the rate of mutation per year. The bottom line is this: if you're very short-lived, you're going to have lots of mutations. And if you're very long-lived, you're going to have rather fewer mutations. Now, what does this mean? The actual rate of mutation is probably pretty similar, what you might call the primary rate of mutation, errors. The thing is, with humans, we invest a lot in DNA checking, DNA editing enzymes, systems that go and check that that copy is a faithful copy. Error. Now, that's a timely and metabolically costly process. If you're a mouse, you're going to die at the age of 2 anyway. There's no point in investing in those processes because you're going to be dead, right? So an inverse correlation between mutation rate and lifespan. So what does that mean? This is kind of a cool way to demonstrate it. You've got enormous range in these animals in their size. So we've got a 40,000-fold variation in size. We've got an enormous range, a 30-fold variation in predicted lifespan. We've got, therefore, a pretty big range in mutation rates. But look, this is the end of lifespan burden of mutations. It's only a 3-fold variation. In other words, and there's a nice illustration to make the point. In other words, a mouse gets to its 4,000 mutations or whatever very rapidly. You get to the same end point, but much more slowly. See the key thing here, that the major determinant of net after correction mutation rate is lifespan. Now, how does lifespan relate to size? Obviously, larger animals tend to be longer lived, and vice versa. Longer lived animals tend to be larger than smaller ones. But it's a pretty messy relationship. So here's a log-log plot. This is longevity of a species in years, and this is its size, its body mass. And there are relations. Blue is birds or volant flying, and sorry, yeah, it's not just birds because it's bats as well. And red is terrestrial. Yes, so there is a positive relationship, but boy, is it messy, and boy, are there some weird departures. I take this guy. This is a bat. It's a bat which is this big. It's been recorded as living 41 years. Okay. And this is, we've already mentioned it, the naked mole rat is this weirdly long-lived. It's subterranean, so it sort of hangs out underground, doesn't get in anyone's way. So even though there's a correlation between lifespan and size, it's not a strong one. And in fact, we can sort of unpack the data from this paper a little bit more and sort of realize that the lifespan size thing is sometimes completely irrelevant. So for example, this little guy, the world's ugliest organism, look at it, is the naked mole rat. And this is a giraffe. We all know what giraffes look like. They have rather similar, in terms of the measurement in this paper, similar rates of mutation, right? And yet, obviously, they're completely different in body size. And yet they have more or less the same life expectancy. So they last about 24, 25 years, both of them. Okay. So in other words, it does seem that life expectancy, lifespan trumps size in this mutation rate argument. And by the way, just to make the point, obviously this is not to scale. A giraffe is huge and this is rather small. Let's actually change that and make the point that these really are different sized organisms. There is a naked mole rat in severe danger of being trodden on and crushed to death by a giraffe. And that's not the only data you can point to. Cows and horses live shorter than we do, but they're much bigger than we do. They have many more cells than we do, but they have higher net mutation rates than we do. So the take home on this paper is that mutation rate is, as I said, a negative function of lifespan, meaning that the longer you live, the more you invest in mechanisms that weed out mutations, that prevent mutations, that edit mutations, that screen out mutations. But you know, that's not enough, because elephants and whales are long-lived, but even given the extent to which being long-lived means you've got reduced net mutation rates, that's not enough to prevent elephants and whales getting cancer. We can't explain Peto's paradox solely in terms of this mutation inverse relation with lifespan. So there must be more going on in elephants and whales, and maybe in some other species as well. So that's what I'm saying. The final thing I want to talk about is large body size. There's something more than just this mutation rate relationship going forward. And I've got sort of 3 examples. The first of which, and again, I apologize for just plonking down the abstract of this paper, but it's kind of cool. The focus here is on telomerase. Telomerase is the enzyme that adds bits to the ends of chromosomes when chromosomes are replicated. Okay, there's this sort of, I've never really understood why it has to be there, but there is this fundamental flaw in the DNA replication process, such that you lose the end of things gradually over time for each phase, each round of replication. Telomerase is a clever system that prevents that from happening. Now, normally cells which don't have telomerase, the chromosomes get shorter and shorter and shorter until they reach a point where basically inviable, and that's the end of that cell lineage. So that's what we might call long-term programmed cell death. Good. That's a good thing, because that means they're not going to go crazy and cancerous. So most or long-lived organisms like us, your adult cells are not producing telomerase at all. Yes, early in development, maybe in producing gametes, telomerase is active, but not later in life. It's not true in small organisms. And this is a comparative study of rodents, where you've got quite a range. Think of the largest rodent, the capybara, and a little mouse. And you can, this is body size range. So that on the x-axis, you've got the mass of the species, and this is a metric of the amount of telomerase being produced. The bottom line, and this isn't just true of rodents, it's true across mammals, that if you're big-bodied, you switch off telomerase production. If you're small-bodied, it doesn't matter, you keep it switched on. Why does this matter? Well, one of the key features of many cancers is if I get cancer, one of the features of that cancer is probably turn telomerase on. So even though it's switched off in a, quote, healthy tissue, it's switched on in an unhealthy tissue. So this telomerase regulation is clearly a body size related adaptation to avoiding cancer. There are others which are more specific to particular species, and I apologize again for this great slab of text, but it is an opportunity to stare lovingly at this world's ugliest organism. It has been described as a penis with teeth, which is a little unflattering, I feel. These things live underground, naked mole rats, and they are extraordinarily long-lived. They last a very long time. And that's presumably because, go back to thinking about the glasses, the beer glasses in different bars, they living underground, they're in a very, quote, stable environment. There are very few extrinsic threats. So they've been able to evolve a long lifetime. But that means they've got to evolve the wherewithal for preventing things going wrong, courtesy mutation and producing cancer. And they've produced a number of interesting mechanisms, which I'm not going to go into, partly because they're highly technical, partly because I don't entirely understand them. But the big message here is that a feature of normal healthy cells is they have contact inhibition. Basically, when two cells are growing, they meet each other and they touch each other, they stop growing. That's it, that's enough. And a feature of cancer cells is they lose contact inhibition and start growing all over on top of each other and so on. Again, dysregulated cell growth. These guys, naked mole rats, have all sorts of interesting mechanisms that actually make them super sensitive to contact inhibition. In other words, they're super sensors of contact inhibition, which means there's less likely to evolve the lack of contact inhibition, which is a key feature of the transformation to a cancer cell. But let's finally get to elephants. And I'm going to talk briefly about probably the most best known, the most canonical of all the tumor suppressor genes that have been cataloged today, called P53. I've already shown you this slide showing the cascade of mutations which can result in colon cancer. And one of these steps in this textbook example is a mutation of P53 gene. P53 is a key player in the regulation of the cell cycle. I'm not going to go into it chapter and verse. It's one of these things which is involved in so many processes. It's actually kind of mind-boggling. It's at this nexus of all sorts of networks of critical processes. This gives some sense of it. It's important. And a very large proportion of human cancers have mutated p53 genes, because that's what allows the dysregulated, the crazy, the anarchic cell division to occur. There's no longer that check in place imposed by p53. So p53 is important in cancer. Now, how many copies do you have? How many copies do I have? I have one, well, I have 2 copies, one from my paternal genome and one from my maternal genome. So if I happen to pick up a mutation which knocks out one in one copy, and then in that same tissue, I pick up a second mutation that knocks out the other copy, in that tissue, my spleen, let's say, I have no working p53. And suddenly the door is wide open to the development of cancer of crazy dysregulated cell growth, okay? So if you've only got one copy, then you're kind of on a knife edge in a sense. So what has this got to do with elephants? This is a survey, a phylogenetic survey, as you can see, because this is the family tree of vertebrates, so a large number of species. What we have is the purple dots are the count of p53 genes in the genome, okay? Humans have 1, as I've just said. Now, there are a few species that have 2, and there are a couple which have a few more here. Look at this. That, ladies and gentlemen, is the elephant genome. It has 20 copies of p53, 20 copies. And what's more, we know this is a function, or it's correlated with the evolution of large size in elephants. Elephants' closest relative, as you maybe know, sort of anomalously, is this guy. This is a rock hyrax, okay? That is the closest relative, sits side by side elephants down here, okay? And just, again, let's put that into context. That's to scale. This is a tiny little, relatively short-lived organism. The elephant has this wealth of extra copies. Now, it's a little bit complicated. They're not all strictly functional, and they're still working out exactly what they do. But one thing seems clear, that if you've got extra copies of this key player in the regulation of cell cycle dysregulation, and mutation is occurring, then having extra copies is your friend. You've got redundancy on your side. So this looks like an evolution, body size specific adaptation which has occurred in this one lineage, which is one of the reasons why elephants don't get cancer. So to wind up, so here we are, the p53 genes. We're not sure what's going on with whales. That's a bit more challenging even than elephants to work with. But we do know from studies of cell behavior that elephants are particularly attuned to signaling apoptosis, so telling a cell which is misbehaving itself to go off and die, programmed cell death, okay? Probably related to this p53 regulation. Now, why am I telling you this? Why do you care? Well, you should care. Cancer is, as you know, one of the major killers in modern humanity. If we can draw upon what the natural world has done over eons, this is natural selection doing its work in different species, and these are some I haven't mentioned, but here's our whales, here's our elephant with p53, here's our naked mole rat with its curious systems, and we got other candidates as well. Animals which, if you like, to some extent at least, have solved the cancer problem. And by learning from these guys, I would say, the hope is that we can develop useful therapies. We can learn how to transfer their evolutionary wisdom, for want of a better term, to transfer their evolutionary wisdom to humans, to our species. So this isn't just weird natural history. Oh, aren't elephants wonderful? Hopefully, this will open avenues of anti-cancer research for years to come. With that, Thank you very much. What I'm going to do is drop the presentation. Just a quick highlight. That's my email address. If you have questions, obviously hang around and ask questions. As I'm about to kill the screen share. But if you don't want to ask questions in a public forum or whatever, it's always a pleasure for me to hear from Sabanci people, Sabanci students, well, from Turks in general. And before I sign off, I just want to say, let's hope that round 2 of the election goes better than round 1. Thank you.